Good morning all. I am Dr. Dhondiba, Assistant Professor of Chemistry, Government First Grade College, KR from Bangalore. Today I am going to discuss about 24th session of Biochemistry, which is Chemistry 8th, Paper 8th of which belongs to Enzymes of 6th semester BSc. In this session, I am going to discuss about allosteric enzymes, its definition and example. Then enzyme inhibitions, they are competitive, non-competitive and uncompetitive inhibitions with one example for each that I have to discuss one by one in this session. Now first of all, what we have to know about allosteric enzyme. Allosteric enzymes are the enzymes that have a additional binding site for a effector molecules other than active site. Enzyme, the allosteric enzymes, they having a other than active sites. It is having a other additional binding site will be there other than active sites. The binding brings about the conformation changes. Conformation with that is a lock and key model or induced fit model. This will be when it is complex will be formed. No, that conformation will be changes due to the presence of the allosteric enzymes because of it having a additional binding site along with that is a active site. Therefore, the changing its catalytic properties due to having a these sites present on that is the enzymes, it gives that is a binding that is a binding way or the conformation or structure of the enzymes will be changes and hence its catalytic property will be changes. The effector molecule can be as an inhibitor or activator. That effector molecule no, that is present and that having a some extra additional active sites will be there. They will be a activate the accelerate the rate of the reaction or that is decreases the rate of the reaction. Therefore, it enhances the rate of the reaction or it inhibits the rate of the reaction. Inhibition means it is decrease the rate of the reaction. Activate means it increase the rate of the reaction. Therefore, we say that is a allostery is a process of enzyme regulation where binding at one site influence the binding at subsequent sites. Called that is a allosteric. Allostery is a process of enzyme regulation where binding at one site influences the binding at subsequent sites because of additional binding sites will be there in the enzymes. Now come to that is the examples. Now first example goes to here. I have three examples I have to discuss here. First example goes to aspartate transcarbamylase. That is it is ATCase means it is enzyme because AS is ends means it is a enzyme. Aspartate transcarbamylase, it is ATCase, catalyzes the biosynthesis. Biosynthesis means in the biological path the pyridine will be produced. Biosynthesis of the pyridine, the cytidine triphosphate, that is CTP, cytidine triphosphate is the end product. After that is it gives that is a cytidine triphosphate as a end product and also inhibits the reaction. It also inhibits the reaction. It decreases the rate of the reaction. It is known as a feedback regulation. ATP, that is a adenosine triphosphate. It is a purine nucleotide, activates the process. This formation of the CTP, that is cytidine triphosphate, it accelerates the rate of the reaction by using a ATP and it because it having a extra additional sites will be there and high concentration of the ATP can overcome the inhibition of the CTP. Because of high concentration, the make that is a rate of the reaction decrease and it becomes a end product. It making a end product. It cannot be converted into other product. It is a particular compound. Up to that it make that is a rate of the reaction. Therefore, it is controlling. It regulates the reaction. This ensures that the synthesis of pyridine nucleotide when a high concentration of the purine nucleotide, that is such as ATP, is used, then it is formed, that is a cytidine. Otherwise, the instead of that is a cytidine triphosphate, it will be formed, that is a other product, it undergo a breaking like that. Therefore, it regulate the rate of the reaction, neither fast nor slow. It make that is a expected product. It make that is a expected product, that is called as a allosteric enzymes. Then second example, glucokinase plays an important role in the glucose homeostasis. Homeostasis. It converts glucose to glucose 6 phosphate. It is plays a important role, that is a homeostasis. It convert glucose into a glucose 6 phosphate and enhances glucose, that is a glycogen synthesis in the liver. It first convert that is glucose into glucose 6 phosphate, and it is further it is a synthesis of the glycogen in the liver. It also senses, it is the concentration of the glucose for the release of insulin from the pancreatic beta cells. It is also, that is a make it is a concentration, that is a glucose for the release of insulin from the pancreatic beta cells. The glucokinase has a low affinity for glucose, so it acts when the more concentration of the glucose is present in the liver. Here, glucokinase has a low affinity for the glucose, so it acts as a more concentration for the glucose present in the liver, which should be converted into glycogen. Therefore, the activity of the glycokinase is regulated by the glucokinase regulatory protein. Therefore, it is make the regulation. The product will be formed or not formed, that will be a becomes, that is a glucokinase. Third example, acetyl coenzyme carboxylase. Acetyl coenzyme A carboxylase. It is again a allostatic enzymes regulate the process of lipogenesis. Lipogenesis means a conversion of adipose tissue into lipids. That is a lipid will be a formation. No, lipids will be formed in this process. This enzyme is activated by the citrate and inhibited by the long chain acyl coenzyme molecule. Acyl coenzyme A molecule such as palmitoyl coenzyme A, which is the example for the negative feedback inhibition by the product. Acetyl coenzyme A carboxylase is also regulated by the phosphorylation or dephosphorylation controlled by the hormones such as glucagon and, that is a epinephrine. The production of that is a the phosphorylation or dephosphorylation of the controlled by the hormones such as glucagon or epinephrine by using a allosteric enzymes. These are about, that is a example for that is a allosteric example. Now just I take, that is a, what are the, that is a effector, that is example I put here. Here halokinase and metallic pathway here takes place in glycolysis. Halokinase is enzymes, and metabolic pathway is nothing but it is a degradation will be takes place in the glycolysis. And inhibitor means it decreases the rate of the reaction during the process. That is glucose 6 phosphate is act as a, that is a inhibitor. It decreases the rate of the reaction. It controls the rate of the reaction. It regulates the rate of the reaction. Next, the phosphofructokinase. Phosphofructokinase is an example. That is, it is present in the again glycolysis process, and ATP is as an inhibitor and activator. It inhibit means it decreases the rate of the reaction particular, but activator other, that is activator effector molecules will be there, that will be act as a A AMD means adenosine monodiphosphate, monodinucleotide, sorry, adenosine monodinucleotide. Then here adeno, that is what is that? Adenosine diphosphate. This is the another example. These are the activators. It activate the rate of the reaction. Then third one, isocitrate dehydrogenase. That is, it is present in the metabolic pathway, that is the Krebs cycle, and ATP is as an inhibitor, and ADP, adenosine diphosphate, and NAD means it is a NAD, usually that is a nicotinamide adenosine diphosphate. It is a NAD, which is act as a activator. It activates the, enhances the rate of the reaction, but ATP is decrease the rate of the reaction or inhibit the rate of the reaction. Then acetyl coenzyme A carboxylase is the enzymes. It present in the metabolic pathway like a fatty acid synthesis, and palmitate as an inhibitor, and isocitrate as an activator. And these are the example for that is a allosteric enzymes. Just I have taken that is in, these are the three example for that is the explanatory type. Otherwise you have to be able to write, these are the, you have to take that is a, which one is the inhibitor and it is the activator. It having, because of the enzymes other than active sites, it additional binding sites will be there, and that due to additional binding site inhibit or activate the rate of the reaction. That is called as a allosteric enzymes. Now coming to that is a enzyme inhibition. What is mean by enzyme inhibition? Now many substances can reduce or eliminate, or reduce or even though eliminate. Inhibition means stop, or it is say that is a decrease. We say that is a removed or stop or decreases the rate of the reaction. Therefore enzyme inhibition, a substance, those substances which are reduces the rate of the reaction or eliminate or stop the rate of the reaction, catalytic reaction, called that is a enzyme inhibition, enzyme inhibitor, and the process is called as a enzyme inhibition. It is a process in which the some substances we are added, some that is the enzymes we are added, it eliminate the rate of the reaction, or it is stop the rate of the reaction, or decrease the rate of the reaction at particular specificity of enzyme, and that is called as a enzyme inhibition. These substances are called as a inhibitors. Inhibitors means those substances which reduces or eliminate the catalytic activity of specific enzymes. Catalytic activity. What is the catalytic means? It is make the accelerating, where binding sites will be there, and that binds with the substrate, and the rate of the reaction will be increases. The catalytic activity of specific enzymes will be a decreases, and that is called as a inhibitors. Inhibitors are a compound that bind on a enzyme and prevent either formation of enzyme substrate or break down the enzyme and product. Therefore inhibitors prevent, it cannot allow to find the binding properly. When the enzymes react with that is a substrate molecule, it is binding will be takes place, it complex will be formed, and then it is a breakdown, and it is the rate of the reaction enhances. This will be happen in normally. But when the inhibitors are present, the inhibitors it having it prevent the binding. Binding it cannot be allowed properly. It binds some, and therefore some molecules will be bind, or some active centers will be a that is a bind with that is substrate molecules, and the rate will be a goes on. But it prevents some that is a reducing, and it cannot be allowed to form a enzyme substrate complex, or if it is formed, it will immediately break down that is enzyme, and that is a product. It will be a, this like that is a process will takes, that is called as a inhibition. Now coming to the inhibitor may be, that is a inhibitor may be reversible or irreversible. It may be participate in the reversible reaction or irreversible reaction. In irreversible reaction, irreversible inhibitors like iodoacetate and the antibiotic penicillin usually bind the covalently to one of the side chain group of the active site, thereby eliminating the enzyme activity. When the inhibitor combined with that is a particular substance at particular group, their activity will be a remains that is a retarded. Therefore, we say that is a so many examples will be there. So many examples will be there. If it is cis form, that is cisplatin. For take the example, it is a coordination compound cisplatin. It is a active for the treatment of the anti-cancer reagent, anti-cancer. It is act as an anti-cancer drugs, or it is a treatment for the cancer. Cisplatin is used, but whereas transplatin is a it is a inactive, remains inactive. It is a not at all active. Like that, no. Therefore, we say that depends on that is inhibitor. That is a combining. When inhibitor combined with the particular that is group, it the particular group is not available, and that group will be becomes inactive. Therefore, the activity of that enzyme will be retarded or stopped, and such that is called as it is also it is takes place in reversible and irreversible process. Now, in many nerve gases and pesticides are irreversible inhibitors of the vital enzymes for nervous systems. Reversible inhibitors may be that is reversible. These are the irreversible. Irreversible means it cannot be reverse takes place, and then reaction will be takes place. Many nerve gases and pesticides are irreversible inhibitors of vital enzymes of nervous system, whereas reversible inhibitors may be easily removed by the methods of dialysis. Dialysis, dialysis means that is purification of the blood. A process it is a make that is a, in the kidney now. The purification of the drugs will be that is a purification of the bloods will be takes place. That is called as a dialysis from the enzyme solution. Therefore, reversible inhibitors may be easily removed by the method of dialysis from the enzyme solution. Now, coming to that is the types of the reversible inhibitions. There are two common types of the reversible inhibition. Along with two, one more will be there. Just try to say that is the three types of the common reversible inhibition. First one is goes to that is a competitive inhibition. Competitive inhibition. In this inhibition, the substrate, the structure of the competitive inhibitor, resembles with the structure of the substrate enzyme. Now, substrate enzyme will be there, and the competitor inhibitor molecule will be there. The structure will be similar. They are having a same structure. Then competitive inhibitors are a substrate analog. Therefore, we say that the competitive inhibitor molecule having a structure similar to the substrate enzyme. Therefore, the competition will be takes place. Therefore, they are complete. That is a compete with that is a substrate for the active site, blocking it by the not reacting. Therefore, here the enzyme having a there and substrate molecule will be there. Substrate molecule having a same structure with that is a competitive inhibitor. Therefore, enzymes having a particular active centers. The inhibitor also has a capacity to bind with that is a enzyme active sites, and substrate molecule having a also capacity to bind with that is a enzymes. Therefore, both are competitively bind with that is a enzymes. Enzymes at active sites are active centers. Therefore, the blocking of that is active sites will be a takes place. Therefore, it is a drags or it remove there is a active centers which are available for the substrate molecule to increase the rate of the reaction, or reaction will be catalyze the rate of the reaction. But when competitive inhibitor will be there, that competitive inhibitor will be attack on that is the active centers of the enzymes. The availability of the active centers, active sites to the substrate molecule decreases by blocking, but not it is a reacting, and hence the rate of the reaction will be decreases, or it is almost all stop, or it is the rate of the reaction decreases. Therefore it is called as a competitive. Therefore the substrate molecule structure resembles with the there is a inhibitor molecules, and therefore substrate and inhibitor having a competition to drag that is the active sites from the or react to the active sites of the enzymes, and the reaction will be takes place. If the inhibition, that is inhibition molecule competition will takes place, more the inhibition molecules block the active sites, therefore rate will be a decreases more. If less that is a blocks or active sites will be a not free for that is a substrate molecule, then it will be a rate of the reaction will be a that is a decreases. Therefore it is being may takes place by blocking it by but not reacting. Example here, the melanate competitively melanate competitively uh, competitively inhibits the enzyme succinate dehydrogenase, which convert the succinate to fumarate. Succinate to fumarate. These are the succinate and fumarate. Succinate and fumarate. These are the only transform. That is a fumarate is the transform and succinate is nothing but cis form. This is cis form, and that is a fumarate is nothing but it is a double bond will be there, and it is a transform. Actually transform, that is a isomer which will be takes place. Okay. Now here the melanate will be act as a competitive inhibitor, and it is resemble the same structure, like this is the same structure, the COO, CH2, CH2, and this will be a, takes place here. The melanate, actually this will be a, not actually it is wrong one. This is the melanate means it is a COH, CH2, and this is the only COOH. That is a COO minus. Therefore H2 is the extra here. Therefore COO that is CH2 and COH. And this group, these are the similar to each other, and only one group will be a methyl group will be a that is extra here, melanate. That is it is melanate is comes from that is the melanic acid COOH, CH2, COH, and succinic acid is nothing but COH, CH2, CH2, COH. That like okay, and when it is these are having a competitively block that is active centers of that enzymes, and it gives that is a fumarate, and rate of the reaction is decreases, and this is the happening will be takes place by that is a competitive inhibitor. Then similarly benzamide competes with the arginine. Arginine reduces of peptides for the active sites of trypsin. The active sites of the trypsin will be there. Then benzamide it competes. Benzamide plus arginine. These are the this is the substrate, and benzamide is act as a competitive inhibitor. It compete with that is arginine to in the peptide bonds, and it blocks that is the active sites of the trypsin, and it is a form that is a product. The rate of the reaction will be decreases or stop, whatever may be. It is the element. Now come to that is sulfur drugs. Sulfur drugs are another example for due to the similar structure of that is the para-amino benzoic acid. This is the para-amino benzoic acid. That is a para-amino benzoic acid. It is a similar structure of that is a sulfur drugs will be there, and therefore it is essential to the vital bacterial enzymes. And the effect of competitively inhibitors can be reversed in increasing the substrate concentration. The people with methanol, no, methanol poisoning are the treated by the administering the ethanol. Now here the pure ethanol is the it is fit for drinking purpose. You are added the water and it make dilute, and they take for that is like a beverage. No, beverage means it is a drinking purpose. It is drinks there will be having a, but it having alcohol, ethanol is the 100%. It making the dilute, and that will be it is a fit for drinking purpose. But when we are adding a that is a competitive inhibitor like a methanol, and it make there is a poisoning the whole ethanol. Therefore, methanol, methylated methylated ethanol is a, it is a poisonous, and it is a not, there is a drinkable. Therefore, here it is called as a competitive inhibitor. Here methanol is a competitive inhibitor or alcohol dehydrogenase, whose substrate is ethanol. Ethanol will be a substrate, and alcohol dehydrogenase is nothing but it is a enzyme, and it blocking the competition between the ethanol and methanol, the rate of the reaction, and therefore it is became a poisonous. And this is the happening in the competitive inhibitors. Now here what happen here usually, the enzyme, that is a E, and substrate will be there. When enzyme plus substrate, there is the absence of the inhibitor to form, there is a ES, that is a activated complex, that is complex, that is a like enzyme substrate complex, and it will be a breakdown and to form that is a enzyme and product. But when this inhibitor will be there, inhibitor and that is substrate molecule, they readily attack on that is the enzymes, because suppose we say that is the enzymes having the active centers or active sites will be 100, then substrate molecule can be occupied, that is 60 or 50, many it is attack, and some of the that is the active centers or active sites will be occupied by that is the inhibitor, and therefore this complex will be formed also. Therefore, the full 100% that is the formation of active cell, that is a complex, no, 100% formation of enzyme substrate complex is retarded, it is prevented, it is stopped or eliminated by blocking that is a EI. Therefore, it inhibitor binds with that is the enzyme active centers, and it became a enzyme that is inhibitor complex, and due to the less number of that is the availability for the active centers for the substrate, and it form that is the complex that is the enzyme substrate complex, and which is further breakdown and to form that is a that is product as well as enzyme. Therefore, due to that is a blocking, the more is the blocking, the rate of the reaction is the more decreases. Less is the blocking, less that is the active centers will be blocking by that is inhibitor, the less rate of the reaction will be decreases. And this is the happening by that is a competitive exam, because competitive, then in competitive the structure of the inhibitor and that is substrate inhibitor will be a synonymous, similar. It is therefore they are both, they can be occupied on the active centers directly. If it is not similar, then chances for inhibitor, it cannot be allow the enzyme to attack that inhibitor. This will be happening. Now come to that is a non-competitive inhibition. Non-competitive inhibition. In this inhibition, the inhibitor binds to the enzyme at a site away from the active sites. Now in this inhibition, the inhibitor binds the enzymes at away from the active centers. Suppose suppose a filter paper will be there. In filter paper having so many, there is a porous will be there. Other than that is place of the porous, the other space on that is the enzymes will be there, or filter paper will be there. That will be available for, there is space for available for that is a binding the inhibitor. That is called as a non-competitive inhibitor. Okay. Therefore, these are not substrate analog. These cannot be structurally related or synonymous structure with that is a substrate molecule, and can bind either to the enzyme substrate complex. Therefore, it is not there. When first the substrate molecule combined with that is enzyme molecules by active centers, first it formed that is a complex. Then at that complex, this inhibitor combined, and therefore decomposition rate will be decreases, and the rate of the reaction ultimately decreases. This causes the change in the three-dimensional conformation of the enzyme, preventing the formation of reaction, preventing the reaction from occurs or to reaction occurs. Suppose complex will be formed. If it is further complex cannot be decomposed, then there is no product. Then complex that is reversibly comes to that is again reactant. First they bind whatever the this non-competitive inhibitor this is bind away from the active centers or active sites of the enzymes. Therefore, this is the free available for the substrate. Substrate molecules easily bind with the active 100% active sites of the that is enzymes, and a complex will be formed. But here the inhibitor because it is present along with that and it is other than active site, it binds no, therefore it forms that is it is present at the complex, and further the decomposition of complex is prevented. Therefore decomposition of the complex prevented means again decompose and it get that is back to the reactant, and the rate of the reaction or the reaction to occur is prevented. Therefore a rate that is a rate will be that is a reaction will be a produced, product will be formed, or it is formed then it is slow one, rate of the reaction will be decreases, and this will be takes place by non-competitive inhibition. Now come to there is a example for this. The AMP, that is a AMP, it is actually adenosine monophosphate. It is a non-competitively inhibits the enzymes fructose 1,6-diphosphate phosphatase. It is an enzyme. It is non-competitively combined with which convert the fructose 1,6-diphosphate to fructose 6-phosphate. It converts fructose 1,6-diphosphate to the fructose 6-phosphate. Once for one phosphate, there upon one position, the phosphate group will be there. That will be removed by that non-competitive enzyme. There is a non-competitive inhibitor, and it convert that is a fructose 1,6-diphosphate to fructose 6-phosphate, and Pestatin as a non-competitively inhibits the enzyme rename. That is another that is enzyme actually Pestatin. Pestatin is the another enzyme. It is non-competitively inhibits the enzyme rename. And this is the example for non-competitive inhibition. Now the effect of the non-competitive inhibitors cannot be reversed. Cannot be reversed by the increasing the substrate concentration. Suppose we are increasing the substrate concentration, the rate cannot be reversed. Cannot be reversed. Now here in this note, the enzymes will be there already, the substrate will be there, the 100% that is the active sites will be free for that is combined with, but it is reversibly enzyme has the ability to bind with the enzymes at the space other than active sites. The space on surface of the enzymes were away from the active sites, and it is a complex form. The enzyme inhibitor complex will be formed. Then this is the complex will be formed. Then it is conjunction will be takes place. Then the substrate molecule will combined. Then it formed that is a here enzyme inhibitor substrate complex. Then it is a breakdown further. Then it formed that is a enzyme substrate complex. Then further it is decomposed to form that is a enzyme end product. Or this is the directly, and this is the in options of that is a inhibitor. It will takes, but in presence of that is this will be takes place by this type reaction. First it is a inhibitor directly react with that is a enzymes. Other away from that is a enzymes active centers, active sites, and then the remaining that part when after there is a along with that is the enzymes with inhibitor substance, they further combine with that is a substrate molecule and the form a complex there is a enzyme inhibitor, there is a substrate. This further it is a decompose to form that is a only it is a decompose and eliminate that is a enzyme inhibitor if it is takes place, and the enzyme substrate complex will be formed, and further it is decomposed and the form the product. Otherwise it immediately breaks and it back to the reactant. It goes to that is a, suppose concentration will be increases, it cannot be reversed. It cannot be reversed. It will be a takes place, and here it is the this type of the reaction is called as a non-competitive inhibition. Non-competitive inhibition. Now next come to that is the last one, that is the third one, uncompetitive inhibition. Uncompetitive inhibition. This is the third type. This is the third class of the reversible inhibition, which however it is not very common. Usually it is rarely occur. It is not common, but it is a reversible inhibition where in this case the inhibitor does not bind with enzyme but only binds with the enzyme substrate complex. Therefore here usually first the inhibitors it is followed that is a it attack on that is a complex but not either in the enzyme or substrate. First the enzyme combined with that is substrate to form that is a complex. At that complex the inhibitor attack and it make that is a the enzyme uh, enzyme substrate inhibitor complex or enzyme inhibitor substrate complex, and this will be a make that is a rate of the reaction decreases. It will be formed this again due to this is the extra formation and a less amount. Now whatever will be not affect, the complex cannot be bind the inhibitor, that is a enzyme and substrate complex, which one how much it is not bind with that is a inhibitor, that will be decomposed and form that is a product, and remaining that will not be formed. Therefore the rate of the reaction will be a decreases. Higher the product, the rate will be a increases. Less the product, that is a it will indicate as a less product, indicate as a rate will be a less or rate of the reaction will be a slow. Therefore it is a extra formation of that is a enzyme inhibitor, there is substrate complex, and this is called as a uncompetitive inhibitor. Uncompetitive inhibitor, this is the only binds with the enzyme substrate complex, and uncompetitive inhibitor decreases both the there is a Michaelis Menten constant, Michaelis Menten constant, and the maximum velocity reaction, maximum velocity of the catalyzed reaction, that will be a decreases in the enzymes. Therefore these terms will be a decreases. Therefore it is ultimately rate of the reaction will be a decreases. And so far I have discussed in this session. Now so far I have discussed that is a competitive and non-competitive and uncompetitive enzyme inhibitions. Now come to that is the differences. It may be asked for that is a in exam for 2 marks. Write the differences between competitive and non-competitive inhibitors. The first one is competitive inhibitor and another one is non-competitive inhibitor. These are the substrate analogs and these are non-substrate analogs. Means the structure of the inhibitor, it is exactly similar to the structure of the substrate molecule. Therefore substrate analog. Here the non-competitive competitive inhibitor, the inhibitor do not having a similar structure of that is a substrate molecule. Hence it is called as a non-substrate analogs. Then it is the second one actually. It is the one is over. It is second. It binds directly to the empty active sites. The competitive inhibitor, it is drags active sites on that is the enzymes, because suppose 100% active centers will be there, out of 100 it make that is a competition in between, the competition of substrate as well as inhibitor takes place to attack on the enzymes and to fill that is the active centers. Therefore it some participates, some that is active centers will be a drag by that is inhibitor, therefore rate of the reaction decreases. Therefore, binding directly to the empty active sites, blocking it by not reacting. It is a active centers blocks. It makes, therefore rate of the reaction decreases ultimately. And next here, it bind at the site other than active site. Non-competitive inhibitor always it binds at other than active site wherever the substrate occupies in it or not. Substrate cannot be occupied or not, but it is a bind other than active sites in the enzymes, and it causes the conformation changes, prevent the reaction from occurring. Due to this reason, it changes the conformation. Active, that is a native conformation will be there, then rate of the reaction will be increases. Otherwise, it product will not be formed. The conformation or the structure or native structure of that molecule will be a changes, and hence the rate of the reaction will be not occur. This is the inhibitor. Then another one is the last one. The effect can be reversed by increasing the substrate concentration. The competitive inhibitor in which the substrate concentration increases, the rate of the reaction will be made reversible. But it is not a reverse by increasing the substrate concentration. This is the exactly opposite. The effect cannot be reversed by increasing the substrate concentration, but it effect will be increased by increasing the substrate concentration. Therefore, this will be happening. These are the differences between that is the competitive and non-competitive inhibitors. So far, I discussed in this session allosteric enzymes. That is what is mean by allosteric enzymes. That definition I have given, and few explanatory examples I have 3 givens. If you not understand properly, then you have to give that is in table whatever will be there. That is binding site will be there. Additional that is will be there. That you have to prepare, and then I have explained also what is mean by enzyme inhibitions, then types of the inhibition, competitive and non-competitive and uncompetitive enzymes with examples I have explained. And so far I hope you have fully understand. If any doubt, you have to comment in comment box. Thank you.